All right. Well, thanks everyone for joining us today. My name is Michael Mena. I'll introduce myself in just a moment. I'm joined today by my colleague, Dr. Onyema Ogbuagu, and today we're going to be talking about COVID-19 vaccines. We're going to ideally, hopefully give you all updated information on where we are with vaccines in the country today, which vaccines are getting out to individuals, and what are some of the safety profiles of these vaccines? What are some of the considerations that physicians should be thinking about when looking at different vaccines and answering questions for their patients or for other individuals that are coming to see them. So again, my name is Michael Mena. I'm a physician and a faculty member at the Harvard School of Public Health in the Department of Epidemiology, as well as the Department of Immunology and Infectious Diseases. I generally work on development of immunological technologies to monitor and track outbreaks and better understand the immune system's response to vaccines and infections. I'm also a physician at Brigham and Women's Hospital, where I help to oversee COVID-19 diagnostics in part this year, but more generally molecular virology diagnostics in the Department of Pathology there. So I want to turn over for a moment to Dr. Ogbuagu to introduce yourself.
Hello, everyone. So my name is Dr. Onyema Ogbuagu. I'm an infectious disease specialist on faculty at Yale and associate professor of medicine. I direct the clinical trials program. So we've been knee-deep in COVID-19 therapeutics, including remdesivir and some other therapies you may not have heard of in the pipeline, as well as running the Pfizer vaccine trials and now doing the phase 2 Sanofi vaccine trial.
Great. Well, thanks so much. Our disclosures are up on the screen. And so I want to point out that this program today is the first in a series of live streams that are going to be generally about vaccines to prevent COVID-19. Really what our focus is again today is providing some of the most updated information on the vaccines that are authorized or those that are in late states of development. And so we both put different tweets out, and you can check those out. They kind of coincide with this seminar. So we will be largely addressing different vaccine candidates and talking again about those that are including those that are in late stages, not yet available in the United States, but getting there ideally very quickly. We'll talk a bit about the safety and efficacy data. And of course, prescribing considerations. So I want to start with an overview now of where we are at the current state of COVID-19 with the pandemic in general, and of course with vaccinations. So we're just coming off of the heels of a massive wave of this virus across the United States. Of course, we had, it's hard to even call these waves. They've blended into each other. But there are clearly three peaks that we've seen. We are clearly on the downswing of the largest one. It started back in October, November. It crossed the United States, peaked about a month ago or so, and has been dropping pretty precipitously up until really just the last few days, when it's starting to appear to potentially be plateauing. Deaths, as we know, have followed these cases. We still are seeing massive numbers of infections today, despite the large reduction. My personal feeling is this, and I think the feeling of most of my epidemiology colleagues are certainly nobody's assuming that this pandemic is done. But we are seeing a bit of a reprieve relative to where we were in November and December and January. Unfortunately, deaths are remaining high. We're still up at still very high numbers of deaths, far too high for any of us to become complacent, particularly as we start to see news reports about large relaxations of mask mandates and other things. But it is at least nice to see that we have seen a large reduction. I think that the vaccines that we're getting out to the communities today are going to benefit greatly by these large reductions. So far, I would say that the vaccine uptake has been increasing. Of course, it's becoming better and better and better every day. So far, we have over 50 million people in this country who have received a single dose, which is tremendous, and just about 25 million who have actually received both doses of their vaccine if they're in a 2-dose vaccine schedule. So in general, this has been a tremendous uptick. I think when we first started to see these vaccines rolled out, we were seeing a struggling distribution, but now we're kind of hitting our stride. And of course, just a few moments ago or earlier today, it was announced that we should be able to have enough vaccines in the United States for every adult by the end of May, is what our president's telling us. So if we look at the different types of vaccines, we started with a huge number of different vaccine candidates. Today, we have, of course, gotten down to a much more selective, a much smaller group of vaccines that have actually made it through phase 1, 2, and 3 trials, and some of them in the US, some of them elsewhere. Of course, the most the first ones were the Pfizer and Moderna vaccines, the mRNA vaccines. And AstraZeneca is not yet authorized in the United States, but of course is in late stages of development to hopefully start being deployed here. And then there's J&J vaccine. This is a viral vectored vaccine, which was just recently announced this week. And so we are going to start seeing this plethora, this whole ecosystem of different vaccines, which on the one hand is incredible. I don't think anyone necessarily would have thought that we'd have such a variety of vaccines a year ago, of course, but it also means that it's a complicated landscape. And a lot of physicians and individuals who are getting vaccinated are just wondering what the heck to make of all of this. So hopefully we will be able to provide some information to you all that's useful. So I'm going to hand it over to Dr. Ogbuagu to start talking, to give us some information on different approaches that we've taken with these vaccines.
Yeah, thank you, Michael. So we'll move on to talking about the different vaccine approaches for COVID-19 immunization. We know that there's multiple different strategies and vaccine technologies and approaches to COVID-19, but we're going to really focus on those that are occurring in the United States. And so the three main approaches that have emerged have been the viral vector vaccines, with the poster child for that being the Johnson & Johnson or Janssen vaccine, as well as AstraZeneca, and the protein subunit vaccines represented by Novavax, as well as the gene-based vaccines, which are mRNA technology that utilized by Pfizer and Moderna, as well as the viral vector vaccines, because they also incorporate the DNA for the spike protein for the SARS-CoV-2 virus. So these have emerged as the approaches that have been utilized to advance some of the vaccines for SARS-CoV-2 approach. We're going to discuss each one of these individual approaches in the upcoming slides. So let's start with the Whole-Microbe Approach. So in general, these include utilizing a virus, the SARS-CoV-2 virus, which can be inactivated, and which means it's really processed to ensure that it doesn't have some of the pathogenic mechanisms, and also can't replicate. Examples of these include flu vaccines, as well as the polio vaccine. These are primarily being pursued through international efforts for the vaccine development process. As you can imagine, exposing individuals to these inactivated microbes are the pathway to which they trigger immune responses. There are also live attenuated vaccines. We know about the measles, mumps, rubella, for example, as well as the varicella and shingles vaccines, which also utilize that technology. In this case, the viruses are alive, but they're attenuated, but they're great to trigger the immune responses. And then we have relatively newer kid on the block, which are the viral vector vaccines. So that's the word. Typically, they are vectors, which means that they really transport or carry some of the gene sequences for the SARS-CoV-2 virus. primarily for the spike protein, and these have been used to deliver genetic material into host cells, and which then eventually result in producing the spike protein, which again stimulates the immune system. Most of you may be aware that at least the US FDA did approve the Ebola vaccine, I think in 2019, which utilized the vesicular stomatitis virus. And so with COVID-19, you can clearly see that different approaches have been used, including the chimpanzee adenovirus by the AstraZeneca Oxford platform, and the adenovirus by Janssen and other technologies. Again, these viral vectors are tweaked so that they are not able to replicate and cause disease when they're administered. The other approach is the protein subunit approach. This is kind of an older strategy for vaccines. And so in this case, either a specific part or subunits of the target vaccine antigen. And we know for COVID-19, it's essentially the spike protein. There are examples of other vaccines that utilize the same approach. Some of them are listed, including hepatitis B and diphtheria vaccines. And so for COVID-19, there are currently platforms utilizing that technology, typically involving spike protein and also adjuvants to improve the immunogenicity. These are platforms being run by Novavax and upcoming studies by Sanofi, as well as other international studies. And then the third approach we will discuss is the genetic material approach, which either delivers instructions through DNA or mRNA. You know that mRNA vaccines are truly a novel approach that were essentially non-existent. Now, the technology had been percolating and studied over a course of decades, since the early 1990s. However, its application to this advanced stage of vaccine development process has really been occurred with the SARS-CoV-2 virus. And so the mRNA is the genetic template that encodes for the spike protein. And, once delivered, encapsulated with lipid nanoparticles to protect it from cellular degradation, it's utilized to generate the spike protein, which again triggers the immune system. Similarly, you could see the viral vector vaccines as well as using this approach, because they also incorporate the DNA encoding for the spike protein, and through which they're delivered to the cells again to elaborate the spike protein, which again triggers immunogenicity. Next slide. So now that we understand the different approaches to the vaccines, let's review the vaccines currently available or in late stage development in the United States. And these are listed under this table. So again, they fall into the three categories: messenger RNA, viral vector, DNA-based vaccines, as well as protein subunits. As you're aware, there are different properties of the vaccines, or delivery dosage approaches that are a little different between them, which I think most of us are aware of. So for the Pfizer mRNA vaccine, is 2 doses given three weeks apart. For Moderna, is 28 days. Both of these require relatively colder frozen temperatures, although they do have decent stability around refrigeration temperature. The Pfizer group have submitted newer stability data to the FDA for review. Based on the age group range of participants who were enrolled in the trials, we know that the EUA, for example, for Pfizer is from age 16 and up, while for Moderna is age 18. As Dr. Mina rightly mentioned, we just got some good news about Janssen over the weekend regarding the EUA, with the advantage and the distinguishing characteristic being that it can be administered as a single dose and can be stored for months at refrigeration temperature. Also, this was studied in adults, so the EUA really just covers adults 18 years and older. We know about the AstraZeneca vaccine, yet to receive EUA in the US, but again, given 2 doses apart, and it's been studied anything from 4 to 12 weeks apart. And both that vaccine and the Novavax protein-based vaccines both require 2 doses and can be refrigerated, and yet to have EUA filing in the United States. All right, so let's now, I know we've all heard about some of the COVID-19 variants and what the implications are for the current batch of vaccines that we have available, and also how they impact vaccine efficacy. So in the next slide, we'll be looking at that. So we all know, and again, the variants have very exotic names, and sometimes based on where they were first identified or other ways that we designate them. But we know the wild type virus, or initially the original virus, if you will, was from China. And over time, we know that the SARS-CoV-2 virus does accumulate mutations over time, albeit at a slow pace, compared to some other viruses like influenza, for example. But over time, there have emerged different variants, and variants meaning that they've had different mutations. Remember that there's a whole host of mutations that do occur as the virus divides. Many of these don't have any significance, but some do. And particularly the ones that have garnered interest are those that are mutations within the spike protein that do result in amino acid substitutions, and then can start to result in potentially conformational changes, as well as impact the way the virus is able to interact with the host cell receptor binding affinity, viral fitness, and then also be able to evade immune responses. And so there will continue to emerge to be variants. I think we should not be surprised that there'll be new variants emerging over time. So many of these have characteristic mutations, which have, the numbers have been described, but paying attention to the mutations in the spike gene. So there's N501, which is carried by both the B117 identified in the UK and the South Africa strain. It appears that for that strain, that specific mutation, it really doesn't impact responses to the vaccine that much. However, there's certain mutations like the E484K, which is notorious for starting to result in decreased neutralizing ability when exposed to both convalescent sera from people who've recovered from COVID, as well as those who are post-vaccination as well, and remain very concerning. So again, the concerns are that these changes in the virus, in some cases, have led to more efficient transmission and reduced antibody binding and immune protections. And this has actually been reflected in some of the efficacy numbers for some of the vaccine candidates that were studied in parts of the world where some of these strains are predominating. And some of the newer ones, like those identified in New York City and California, again, are still being followed for their implications for both diagnostics, disease severity, and ability to evade vaccine-induced immune responses. All right, so then let's want to describe the efficacy of the vaccines, as we very well know. So both the phase 3 studies for Pfizer and Moderna, we know that the Pfizer vaccine trial included about 44,000 individuals. The number was expanded over time to include adolescents who were 16 and 17, as well as people with HIV infection, viral hepatitis that was either cured for hepatitis C or but not causing liver disease. And also the Moderna study included about 30,000. Across both studies, it was pretty clear that you start to see some protection after the first dose and increasing significantly after the second dose. So some decent degree of protection after the first dose, but maximized after the second dose. It was nice to see, reassuring to see that the level of protection was maintained across different race, ethnicities, and age group. Slight dip in the efficacy for older individuals in the Moderna study. But the good news about both vaccine platforms is just how wonderful they are with protecting against severe and critical illness, as well as hospitalization and death. While we don't have, just because of the timing of the clinical trials, that these were the first wave of phase 3 trials conducted before variants were abundant in the sites where the clinical trials were conducted. We don't have direct efficacy data for these vaccines quite yet. However, there's in vitro data for both the Pfizer and Moderna vaccine showing somewhat decreased neutralization capacity, but again, marginally effective for Pfizer and quite a little bit blunted for Moderna, but however not expected to impact efficacy of both vaccines against the variant. And then for the Janssen vaccine as well, remember this was a single-dose vaccine. You are aware that there's a 2-dose trial that's currently ongoing. For the single-dose vaccine, the efficacy was roughly about 66%, with variation depending on the study site. In the US, is about 72%. And we know in South Africa, where there was circulating variants, the efficacy was a little less, as mentioned, about 57%. However, regardless of those differences by circulating variant, you can see that severe disease and hospitalization and death were, kind of robustly, the vaccines were robustly effective, especially against death, even with that single dose vaccine. Next slide. Okay, and this is the AstraZeneca Oxford viral vector vaccine. Again, this was a 2-dose vaccine, 30,000 individuals, and showing that, again, the efficacy after the second dose was about 66.7%. There were a little bit of differences driven by the interval for vaccines, where if the intervals were 6 weeks between doses, it was 54.9%, and for those who had dosing intervals at 12 weeks, it was about 82%. Again, robust protection against hospitalization and death, and efficacy against the COVID-19 variants are as shown. Important to mention that the efficacy against the South Africa variant was really quite poor, even 10% for mild to moderate disease. We don't quite have the data for more severe forms of the disease, but because of this, it led to termination of the clinical trials within South Africa. Okay, and then for Novavax vaccine, I'm sure most of us have seen the press releases. I want to highlight a few things. Again, large phase 3 trial after the second dose. If you look at the point estimate of all, regardless of the strains, it was about 89% protective. But when you looked at the subset that have the original circulating strains, it really is at par with the messenger RNA vaccines with a 95.6% efficacy. Again, the common theme across all vaccines are just, again, the robust protection against severe disease and death. And we can also see that the Novavax vaccine took a hit when it came to the B1351 variant in South Africa, where the overall efficacy was about 49.4%. However, it appeared that that number was driven a little lower by the subset of patients who had HIV infection, and we're going to learn more about that in the future. Okay, so big picture is that, again, efficacy data has been reported differently across the vaccine trials. Primary outcomes have focused on symptomatic COVID-19 illness. And data on serious illness, hospitalization, deaths are really most important. And regardless of the vaccine platforms, it appears that as a composite outcome seems to be robustly achieved by really the front runner vaccine platforms. So to date, no hospitalizations, deaths from COVID-19 or deaths from vaccines have reported following full dose of regimen. So typically after the completion of the 2-dose vaccines, for example, it appears that, there's, again, you rarely hear about deaths from COVID-19, which is just a great outcome. And again, because the data have looked really good among adult populations, the lead vaccine manufacturers, Pfizer, Moderna, Janssen, AstraZeneca, have announced plans to begin testing in children, age 12 to 17. Pfizer has completed the enrollment of 12 to 17. Moderna is ongoing, and Janssen and AstraZeneca will be planning those in the spring. So at this point, I'm going to hand back to Dr. Mina so he can discuss the vaccine safety.
Great. Thanks a lot. There's just a whirlwind of vaccines, becoming available. We're seriously we're clearly in an unprecedented time, to make sense of all of this. It's pretty incredible, frankly, to see it all laid out. So there's rightly so a lot of questions about vaccine adverse events, and both common and serious adverse events. This is always an issue with vaccines. Pre-pandemic, pre-COVID, my life is largely around, or was largely around measles and measles vaccines, and adverse events are one of the biggest issues. In this case, we've seen there's a lot of reporting early on about adverse events, but one of the real takeaway here is we have these different vaccines. We're starting to see real world effects. We're starting to see real world data. The common effects and the common adverse events that we've seen are not all too different from one another. Chills and fatigue and headaches and generally flu-like symptoms have occurred in a large fraction of people, up to 50% of people, for the two mRNA vaccines, the Pfizer and the Moderna vaccine. These are normal. In some ways, could these vaccines have been dialed back a bit to have less of an inflammatory response, for example, that would lead to lower fraction of people with common adverse events potentially. But the important thing is that these are, they are eliciting very, very strong and robust immune responses to the point where we're really seeing, as you just heard, the efficacy is extraordinary for many of these vaccines. There is no free lunch, unfortunately, and so we do see these very common adverse events. There are very small numbers of serious adverse events that have been reported that are not necessarily associated, but temporally they seem to be they're reportable. And so we've seen things, all of them, of course, have to do with inflammatory responses, things like appendicitis and MI, cerebrovascular accidents. We've seen these in very, very, very few numbers of individuals, relatively speaking, 6 per million or 2 per million cases with anaphylaxis. So that's, of course, very serious, and they need to be considered, and that needs to be, we have to be prepared for those, but they are extremely rare. With the Janssen vaccine so far, of course, the data is just now coming in, but very similar effects, maybe not quite as robust a response, but fever and fatigue, some soreness, and in fact, with the Janssen vaccine, serious adverse events were actually seen in the placebo group versus the vaccinated group. If we look at the safety of some of the emerging vaccines, at least on the US market, AstraZeneca and then the Novavax. So AstraZeneca, also very, very similar effects. Again, these are largely due to the immunological responses, which essentially suggest that your body is responding appropriately to these vaccines. There's been a tremendous amount of confusion about this historically surrounding vaccines. Probably most people listening to this are very well aware of what's actually behind these sorts of adverse events. But again, these are expected in a well-performing vaccine, and they essentially suggest that the person is taking to the vaccine. The AstraZeneca so far has not had any severe anaphylaxis, nor has the Novavax vaccine. But again, these were early trials for Novavax. We didn't see any serious adverse events. To manage vaccine-related adverse events, of course, anyone who's distributing and giving injections of vaccines needs to be, we have to be prepared for adverse events to potentially occur. So allergic reactions and anaphylaxis are of course, things that we have to be aware of. And so being prepared to have supplies to be able to treat these types of reactions is crucial. And of course, they usually happen pretty quickly after somebody's actually vaccinated. And so, being aware of that is important. Some individuals will have post-vaccination adverse events, especially some of the more common ones are quite common. If individuals need to take them because of headaches and muscular pain or other inflammatory responses, things like acetaminophen, NSAIDs and antihistamines can be taken. I would recommend that they be taken judiciously, but certainly they can be taken afterwards. Reporting on these individuals are now, I haven't actually looked at this, but I've heard about it. The CDC has this vSafe app to be able to check on patients using smartphones and being able to check in. So we're seeing a whole new group of technologies form to ensure that individuals are able to, appropriately identify when they're actually having something worth reportable, or not. And then, of course, the VAERS system, which is really a vaccine adverse event reporting system, that is one of the mainstays of our ability to monitor post-market monitoring of vaccine adverse events. And physicians, there's usually a pretty low bar to enter something into VAERS. And this is something that's pretty routine for most physicians who are giving out vaccines. So which vaccines do we actually consider using? I think this is a question that a lot of people have, especially the people getting the vaccine. Everyone is asking, what are the vaccines that we should get? And the real takeaway for this is it's pretty simple. The supply chain considerations right now are such that we don't really have too much of a choice. Most people are going to sign up and they're going to get a vaccine that is provided to them. But I think as we start to see more and more information come out about exactly which populations might be best to get one vaccine or another, then we might just start to see some more discerning sort of decisions about who to give what vaccine to. Certainly, when we're thinking about the smarter population level, we can start to look at things like, is this a one-dose vaccine or a two-dose vaccine? What are the freezer requirements? Where are we trying to get these vaccines to? Most places in the US will have access to freezers, but not everyone, and not all places will have access to deep freezers and things like that. And so this has actually been a big issue of how to decide where different vaccines are going and what is the likely follow-up with individuals. For some populations, a one-dose vaccine might actually be a better, a very clear deciding factor, because it might be hard to get follow-up for some people. When we start looking at differences across the different vaccines, if your whole goal is just to prevent any symptomatic infection or severe disease, or when we're considering these different variants, certainly I would say that the data is all coming in now. I think that we are just starting to get a view and a flavor of which vaccines are really going to be effective. And frankly, it's going to be a moving target. It's going to depend on what is the population that you're working with. We just heard, for example, in South Africa, presence or absence of HIV impacts the decisions of potential decisions of how well a vaccine is going to work or which vaccines to use. So all of these pieces have to be taken into consideration. There isn't going to be a very clear answer, I would say, for any given situation. But some of them, like, for example, the AstraZeneca vaccine, may have very good efficacy against, in particular, P1 variant. And so if that starts to become prevalent in a given place, then those decisions might be made to sort of work the vaccine into the community based on prevalence. In the future, we're going to start seeing new vaccines where companies like Moderna and Pfizer are going to start updating their mRNA vaccines to start targeting specific variants as well. And so I think this is going to continue being a very, very moving discussion in terms of which exact vaccine should any particular individual end up getting. So just to summarize all of this has been sort of a whirlwind of information, I feel. Let's see. Yeah, there we go. We've had we've kind of gone through the different types of vaccines that are available. What is the data to date? How might we think about them in terms of adverse events? And in general, we've seen now 2 mRNA vaccines. The Pfizer Moderna vaccines were authorized in the U.S. in December. The viral vectored vaccine was just authorized. And these have all demonstrated very, very high efficacy. As we heard, especially for severe disease and death, they are providing very, very good efficacy beyond what I think any of us really expected a year ago. Some of them are improving on their ability with frozen storage. That was a major point of discussion earlier on. Back in November and December, it started to hit the headlines. How are we going to actually distribute these different vaccines, especially those that are very, very susceptible and require deep freezing for longer periods of time? Now we have things like Janssen vaccine, which can be refrigerated for long periods of time. So access and equitable access is really going to be increasing. One of the major questions, of course, that's on everyone's mind is certainly the variants. How well are these going to do? We're going to have to continue monitoring it very, very closely as new variants. So we haven't seen the last of the variants that are going to pop up by any stretch. I think we are seeing convergent evolution. We're seeing a lot of the same variants pop up over and over and over in different places, which is on the one hand good, because we can start to predict what those variants are going to be and start to act on them. But certainly how well these different vaccines continue to perform in the context of new variants that will arise is going to be something we have to just keep monitoring, and we have to keep monitoring what variants are where and in which populations. We should, my expectation is that we'll see the AstraZeneca and Oxford vaccine, as well as Novavax, coming into the US, and potentially a handful of others as well. So that kind of concludes a lot of our sort of slides, and I want to just open this up now to a discussion with the two of us, and I want to kick it off first. So vaccines are. This is a question for you, Dr. Ogbuagu. Vaccines are touted. They are the absolute focus of our pandemic response. We've had a lot of different foci of the pandemic response at different stages based on what's been available. Of course, masks and distancing testing is something I've talked a lot about throughout this pandemic. And now, of course, vaccines are center stage. Your perspective, is this the end of the pandemic? Are we seeing, Biden's coming out and saying all adults are going to have these vaccines available to them by the end of May? Are we looking at the end of this by the end of May?
I wish I could say yes. So great question. I think that the vaccines have been a welcome addition to the tools that we have, both public health tools and biomedical tools to address the pandemic. Because achieving herd immunity through vaccination, which is the ultimate goal to really reach the end of the pandemic, is going to be challenging on so many levels. We were seeing the difficulties with logistics, although like you rightly said, we're picking up steam. So, for now, I would recommend a comprehensive approach, and all of the above approach for now. As we continue to roll out the vaccines and have more people get them, we still need to maintain those public health measures, so not substitute one for the other, because we know that while we wait for vaccine uptake to take up steam, that masking and testing and isolation it leads to, and just being able to rapidly identify both asymptomatic and symptomatic individuals and have them isolate themselves or quarantine themselves from others will be important to continue the outbreak control. Now, the other part of this is, again, because of the emerging variants and the potential challenges it may pose to vaccinations, I think we all agree that it lends greater urgency to really rolling out the vaccines as fast as we can to really stay ahead of a virus that's clearly mutating and becoming a little more challenging as we get along. The last point I just want to make is that, again, because of the shortage of vaccines, initially, it's been a very challenging issue with deciding who to vaccinate first. And I think if you ask the question as to which group do you vaccinate to have the greatest impact on deaths, will give you a very different answer from which group do you vaccinate to mitigate transmission, right? And I think we've kind of elected to go with the former, which is really target the highest risk individuals, older individuals. And there are many models out there, but again, the convergence of the models seems to suggest that if you vaccinate the older at-risk people in the community, you'll have the greatest impact on death. But again, if you vaccinate some of the more middle-aged, younger demographic, you're likely to have the greatest impact on transmission. So again, welcome additions. I think it's great. I think that the current strategy will convert the epidemic from a lethal epidemic to, if we protect those who are the most vulnerable through vaccination, to, maybe still keeping the numbers up some, but maybe keeping the infections in younger individuals who are less likely to have significant complications, at least until we achieve herd immunity.
Yeah, I think it's a great point. I mean, in so many ways, our response to this pandemic has largely been driven by almost as a medical response versus a public health response, which isn't necessarily well, in some ways, I think it has been inefficient, but especially with vaccines, I think, do we vaccinate to protect those who are most vulnerable, and then, push off vaccinating the ones who maybe are the most responsible for transmitting? I think is a major branch point in the decision-making process for where we vaccinate. And in this case, it actually fits with a lot of us who, as epidemiologists, were saying, back in April and May, if we knew how to protect the most vulnerable people, this actually isn't a particularly devastating virus for the younger individuals by any stretch. And so when balancing, the economic ramifications of staying locked down, social ramifications of that, the last thing we want to do is have the public health response cause any more damage than the actual virus itself would have. So I agree that trying to get to the most vulnerable people first potentially gives us an opportunity to, start to accelerate our reopening scenarios and be able to start to get some sense of normalcy. At the same time, we're seeing people like the governor of Texas today, announcing that all masks are off, and 100% back to normal in the state, which, it's unclear if that's a direct result of the vaccines really becoming much more available, or the reduction in cases, or just throwing caution to the wind and saying, we're tired of the pandemic. So we have these very difficult decisions. So there's been a ton of discussion about the timing of vaccines, and you just mentioned it. We're kind of in a race now. Can we, especially in the context of mutants and variants, which I want to bring up in a moment, but a huge amount of discussion now about how do we accelerate the vaccination of the population. Back in December, I think I wrote an op-ed in the New York Times about trying to immediately do single-dose vaccine studies to see how well the Moderna and Pfizer vaccines would work. And of course, now that's becoming discussed, especially in the context of variants. But what should people be thinking about? I get a lot of people asking questions like, I messed up my appointment, and I have a vaccine. It tells me I was supposed to be vaccinated the second one at 28 days, but now it's not going to be for 35. I guess for anyone who is not so clear on sort of vaccine timing and the spacing, can you give a little background on why these different timing and spacing events were selected, and what it really means from a pragmatic perspective?
Yeah, so remember that we're even talking about timing and spacing of vaccines because there's vaccine shortages, right? If that weren't the case, then this probably wouldn't have come up. And just to say that for some of the vaccine approaches we're using in the United States, so the messenger RNA vaccines, we really haven't studied that question of, delayed second dose versus a much more shorter time frame to getting that done. In the clinical trials, definitely we allowed windows, but the windows are very small, plus or minus 2 days from that. But I think you're asking for much more prolonged durations. I think there's very compelling data from the AstraZeneca vaccine trials that showed that a longer duration between the two doses actually resulted in more robust protection, which was fascinating. But I generally don't generalize until I get more data. So it'd be interesting to see if either with mix and match strategies that are currently being evaluated in the UK, where you either get messenger RNA first and then AstraZeneca first and messenger RNA, and they're studying them at 4 weeks and 12-week intervals to see, answer some of these very important questions about timing. Now, again, the difference between doses would not even come up if we had single dose vaccines, like you rightly mentioned. And I think that's why it's actually great to have Johnson & Johnson vaccine available. I did see some data, or at least some modeling data that looked at deferring vaccinations, waiting for newer versions or single dose versions. And I think that if we decide to hold off on vaccines while waiting for the best single dose vaccine, because we don't want to get a 2-dose vaccine, that can lead to much more hospitalizations due to cases and potentially deaths in the interim. So for now, I think that it's an approach. It's an approach that we really need to learn more about to guide recommendations. So we should allow the science guide the recommendations we make as to what are the appropriate intervals for different vaccine approaches.
Yeah, and the AstraZeneca, the data on that showed actually a potential benefit of spacing, of increasing the spacing. When we take a step back and we ask the question, how does our immune system work? In many ways, our immunity is generally evolved in many ways to see pathogens, not necessarily one right after another, but over time. And so I've actually been very, very interested in that data to understand how our immune system, is it really, the 21 and 28 day intervals, those were chosen effectively to shorten the trial duration. If they added if they made the trials 3 months between doses, we would have had results 2 months later. And so I'm very interested to continue watching that space and understand, do we actually see a reduction in efficacy if we space the mRNA vaccines out longer? I would say that those weren't exactly science-driven, versus just practical decisions to go 21 and 28 days. And so the fact that the AstraZeneca vaccine is actually kind of showing some signs that it improves over time is really very interesting. So something I've been thinking a lot about is in the context of variants and mutations, this concept that, somewhat a similar concept to imprinting or original antigenic seniority. Now, if we induce extraordinarily robust immune responses in individuals with the current versions of the Moderna, let's just say Moderna vaccine for now, and it's inducing a very strong and robust response. And then we want to go in and boost somebody with a variant of the vaccine that has a number of point mutations. I'm curious to get your perspective on the potential for antigenic seniority to kind of kick in and essentially are we running a risk by potentially locking people into an antigenic space, into an immunogenic space? And if we try to boost them with a slightly modified vaccine, is it possible that we might end up seeing that people don't actually end up responding to the point mutations, but just back boost the response to the rest of the protein?
Yeah, that's a very interesting question. I think that we'll have to see. I think certain observations may be instructive. I think that, for example, in South Africa, we're seeing that people who had the original virus, right, tend to really almost be being infected by the newer strains of the virus almost to the same degree as those who were not even infected with the original strains. And so, it'll be interesting to see, after they recover from, their current infection, what trajectories they take regards to a risk of reinfection, etc. I think it's really an open question as to what role the boosters will play. And I think that's what came back to my original issue, as in some people are concerned because of this, that if there's going to be new variants emerging in the future, do I just wait for a new vaccine to initiate a vaccine at that point? And I think that that's a fair question.
Yeah, especially in the context of children. Some people have even gone so far as to say that children shouldn't necessarily even get a vaccine. That we should just, once we have everyone else vaccinated, that we should focus primarily on, continuing to take some mitigating steps, but potentially allowing kids to get infected if that happens, just as kids normally would against the other coronaviruses, which is obviously a very contentious position to take, but one that I think is absolutely actually worth considering in the context of such low mortality in kids. I think some of these inflammatory responses in kids they are very real, but they're also rare, and I think it will be interesting to see how those discussions play out, I think. So do we have any information yet? Of course, you touched on HIV and how certain vaccines are working in HIV patients, but a lot of people have questions about pregnancy, and should pregnant individuals get vaccinated? Do we have any information on how well it's working in both immunocompromised individuals as well as pregnant individuals?
So great about populations that were not necessarily included, especially in the first wave of trials. So for both the Pfizer and Moderna trial, when we recruited individuals, obviously we asked them not to get pregnant, not necessarily because they were anticipated or expected harms, but because that's generally what you do when you do a vaccine study, right? You want to leave vulnerable individuals out of it. So we don't have that much robust data in pregnancy, but that said, there were people who got pregnant during the study, about 20 something or so in the Pfizer and some amount also in Moderna trial. Those individuals are being followed for outcomes. Tens of thousands of pregnant women so far have been vaccinated in the US already. And we know that Pfizer has generated some animal data on reproductive fertility and reproductive potential in animal models, and shown that there really wasn't any impact on reproductive outcomes or fertility. And that Pfizer has actually rolled out a trial for pregnant women with their eligibility criteria being those who are gestational age 24 to 34 weeks. That trial is currently ongoing. So we're going to learn so much more. Again, there were pregnancies on study. There are tons of pregnant women in the US who've gotten the vaccine, and they're now much more structured clinical trials assessing that. However, if we weigh risk and benefits here, in spite of the unknowns, knowing that pregnant women definitely have worse outcomes with COVID-19, higher rates of going on ventilator and dying. So just pregnancy itself as a risk factor, it definitely makes sense for the vaccines that, I would recommend that, women get that, especially women who are at risk of acquiring COVID-19 by virtue of their profession, nurses, healthcare workers, etc. That's really critical. We did include HIV positive patients in the Pfizer vaccine trial, but these were individuals who were on antiretroviral therapy, who had robust CD4 counts. I think the same was also the criteria for the Novavax vaccine trial, which made it even more surprising that HIV ended up being had less efficacy in those individuals. So there's so much more to learn. The number of events in the HIV group wasn't the greatest, so we'll still learn more as the trials evolve. We do know that people with HIV, especially those with low CD4 counts, can have blunted responses. In fact, I don't vaccinate my HIV patients with CD4 counts less than 200 because we know they respond poorly to a broad range of vaccines. So, again, in general, immune-compromised people could have decreased immune responses to the vaccine, but the impact of those on the efficacy of the vaccines, I think we still have much more to learn about.
Great. As we look towards the future, in this country and elsewhere, are there any new technologies and new vaccines that you find most promising, or that you're excited to really see start to enter into phase 1 and 2 trials, and ideally phase 3? I'm curious to hear where your mind goes when you think of exciting new technologies and approaches that we might be able to take.
Yeah. So let's even just start by celebrating the messenger RNA platform, how it can be tweaked. I'm predicting that will be, it's opened like a whole field of opportunities in vaccine. You can probably have a vaccine that you can include, different sequences for different types of viruses. So you can have one vaccine that takes care of either the whole coronaviruses that are circulating, or maybe even potentially other respiratory viruses. So you have one vaccine, or you could have a vaccine and start to address some of the much more challenging viruses like HIV. Maybe have 3 different vaccine antigens in one vaccine construct. The viral vector vaccines also have been a welcome, addition to the group, and it's interesting to see how at least these 2 approaches have really emerged with regards to just SARS-CoV-2 vaccine development. So for me, that's some of the more exciting approaches. There's some much more elegant approaches that are much more early phase development. For example, strategies that are able to utilize innate immunity to protect against acquiring a disease. Some of those are really in early phases, but these are technologies I'm really excited about.
Yeah, this is the interesting piece. I'm happy that you brought up the piece about these mRNA vaccines, that they open up this whole new world of approaches that, as long as the body can translate the mRNA into protein, we could potentially have a single dose with a bunch of different mRNA sequences in it, which does beg the question. When we went and tackled, the global community went to tackle this virus, everything went towards spike, with really no additional effort to include in the same vaccine additional proteins or sequences for those proteins. Looking back, I wonder if we're going to ask, why didn't we think about also adding, other sort of epitopes in there, whether it's potentially T-cell epitopes, or even things like nucleocapsid and other proteins. Do you think that these companies are going to, in the next iterations, are actually going to be just when they go to update their vaccines, are they going to keep the current sequences and just keep adding into one dose, or are they really going to just be kind of taking one out and putting a new one in? Or do you think they'll really go and try to tackle this, sort of have a single vaccine dose, really produce a plethora of proteins?
Yeah, so actually we have somewhat of a proof of concept or something close to that for what you're saying. So I'll give you an example, right? So if you think about the monoclonal antibodies that are evaded by some of these new variants, right? We know the single formulations tend, some of them do poorly. But then when you have, combinations, right, with different targets, for example, like the Regeneron that has 2 different monoclonal antibodies, you can see that it still maintains activity against that. And again, it's spike protein nucleocapsid. So I totally agree. And that's why, like I mentioned earlier, for other, viruses like HIV, where single target, single vaccine antigen strategies have failed, I think it's time to rethink the different vaccine antigens and targets, and maybe like ART constructs for HIV, where you put 2 or 3 drugs together, and if you got the right combination, then you achieved it. I think that's how people should be thinking about vaccines, especially for viruses, that change pretty rapidly, and having more than one target would make so much sense. So I totally agree with your hypothesis, and I hope that informs future vaccine development.
Yeah, no, it's extremely exciting. I mean, it's just it's incredible the innovations that this pandemic has accelerated. It's many of these innovations in vaccines, but also in tests, in so many different technologies. I think that a lasting, there's going to be uncountable lasting effects of this pandemic. But one of the most important ones, I think, is just these technologies that otherwise would have been potentially shelved altogether, or would have taken another 10 years to actually be able to come forward, we're just given an opportunity to shine in a way that, it's not to undercount sort of the negative aspects of this pandemic, but especially when you have so many people getting an infection, it has allowed these vaccine trials to move forward at rapid speed along with the technology, and it's just been incredible to see all of the data flow out of this in all these trials. So it's 8 o'clock, so I want to wrap this up. To our audience, thank you all very much for joining us today. You can claim your CME credits by completing the post-test. You see the link here. I want to thank you, Dr. Ogbuagu, for joining us and for joining me and having this conversation. Any final comments that you want to make?
No, I hope that some of the things that have worked well for combating a pandemic. I was doing a talk earlier today, and someone said, "Hey, for HIV, for example, how come it's been around for 4 decades and you have COVID for 1 year and there's vaccines?" So it's not just about the new technologies, like you said, that were shelved, that were literally dusted off and utilized, but it's about process, how it galvanized the scientific research community, industry partners, all of us working together, literally people who don't play in the same sandbox usually coming together. So, this is really a time to celebrate just, how great things can happen when the best minds come together to tackle a common problem. And I hope that the lessons we've learned from COVID-19 serve us well for future pandemics, which will unfortunately occur.
Yeah. Well, with that, I think we'll close this out, and thank you all again for joining.